This video is about light and how our exposure to natural sunlight and artificial light affects our blood sugar levels. I will share the fascinating science of how light affects our insulin sensitivity, beta cell function, and glucose tolerance, and how you can use this evidence to prevent or reverse elevated blood glucose levels. And by the way, if you think that the only possible mechanism linking light and blood sugar is vitamin D, this video will surprise you. Welcome to Nourished by Science on YouTube. My name is Mario, and on this channel I share evidence-based information about nutrition and occasionally, as in this video, other lifestyle factors, and how we can use the science to prevent chronic diseases. Various factors can cause insulin resistance, beta cell dysfunction, and glucose intolerance. In this video, we will see that lack of exposure to sunlight or too much exposure to artificial light at night can contribute to both insulin resistance and beta cell dysfunction, leading to glucose intolerance, prediabetes, and type 2 diabetes. Sunlight consists of ultraviolet light, or UV light, which we cannot see. Then the range of colors in the visible spectrum, and with even longer wavelengths, we have near-infrared light. Each type of light has a role in helping our bodies regulate blood sugar levels. In this video, I will complete this graph here so that you will fully understand the science of how each type of light affects glucose tolerance and through which mechanisms. At the end of the video, I will give you specific guidance on how to use this evidence to help you prevent or reverse glucose intolerance. Sunlight and glucose tolerance, the role of vitamin D. The first mechanism through which sunlight plays a key role in blood sugar regulation is because sunlight helps our bodies produce vitamin D. Vitamin D is not really a vitamin because our body can produce it. We should think of it as a hormone, or more precisely a hormone precursor. Our skin, when exposed to ultraviolet light, converts cholesterol to vitamin D. That vitamin D is then either stored in our fat tissue or undergoes a series of biochemical modifications, the first of which is conversion to the main circulating form, 25-hydroxyvitamin D. You don't need to know this or all the other complicated chemical names of vitamin D metabolites. Just know that vitamin D itself and also this main circulating form of vitamin D here, 25-hydroxyvitamin D, are inactive forms. So I'll talk about these as vitamin D or inactive vitamin D. However, 25-hydroxyvitamin D can be converted to the active hormone, 1,25-dehydroxyvitamin D. I'll just call this form active vitamin D in this video. This conversion from the inactive to the active form of vitamin D occurs mostly in the kidneys, but also, very relevant to our topic here, in cells of the immune system and also in the pancreatic beta cells, the cells that produce insulin. This active form of vitamin D has numerous functions, mostly through binding to the vitamin D receptor that helps regulate which genes are switched on and off in most cells of the body. It's been estimated that vitamin D regulates the expression of at least 1,000 and maybe up to 6,000 different genes in the human body. What this means is that whether a cell produces a certain protein may partly depend on whether it receives enough vitamin D. Okay, so how is vitamin D involved in the regulation of blood sugar? Well, for one, vitamin D deficiency is associated with insulin resistance. This is partly because one of the genes that the active form of vitamin D helps regulate is the insulin receptor in muscle, liver, and fat cells. In these tissues, the active form of vitamin D directly increases the number of insulin receptors on the cell surface. We need more research on this, but the available data that we do have suggest that people who are vitamin D deficient may have fewer insulin receptors on the surface of their liver, muscle, and fat cells, which would explain why these tissues are less responsive to insulin. Another reason why vitamin D deficiency is associated with insulin resistance has to do with the immune system. Vitamin D can be seen as a regulator of immune function, essential for fighting infections, but also functioning as a brake on an otherwise overactive immune system. Phrased differently, vitamin D seems to play a big role in lowering overactive immune responses. Consistent with this, it has been observed that vitamin D deficient people are more likely to suffer from low-grade chronic inflammation. And that is relevant here because inflammation is a major trigger of insulin resistance. Okay, so we have two mechanisms through which vitamin D deficiency leads to insulin resistance. On top of that, vitamin D deficiency also reduces the ability of the pancreatic beta cells to secrete insulin. The mechanism here is pretty straightforward. The active form of vitamin D is directly involved in regulating how much insulin is made by the beta cells. So if someone is vitamin D deficient, that alone could cause a reduced ability of the pancreatic beta cells to produce and secrete insulin. That's what we call beta cell dysfunction. Well, if vitamin D deficiency causes both insulin resistance and beta cell dysfunction, it would seem plausible to hypothesize that vitamin D deficient individuals are more glucose intolerant and at an increased risk of type 2 diabetes. And yes, we do have pretty good evidence for both as well. On top of that, we have evidence that if we treat vitamin D deficiency and bring vitamin D levels into the normal range, glucose tolerance fairly consistently improves. So the first mechanism through which light affects glucose tolerance is through the stimulation of vitamin D synthesis in the skin by ultraviolet light. There is a lot more to say about this because we can obviously get vitamin D from the sun, as discussed here, but also from food and supplements as well. And you may wonder about food sources of vitamin D and who should consider supplementing, at which time of year, which dose to use, etc. I will cover all of this in a later video, because covering all of that here is well beyond the scope of this video, which is focused on the impact of light. What I wanted to cover here is simply that if you do not spend enough time in the sun, you may well be vitamin D deficient, which could make you insulin resistant and impair your beta cell function, both of which would lower your glucose tolerance and increase your risk of type 2 diabetes. If you are vitamin D deficient, you may benefit from more direct sunlight on your skin. So I have two suggestions. First, if you haven't recently had this done, get your vitamin D level measured and take action if you are found to be vitamin D deficient. Second, seek out the sun more often in a way that is safe and minimizes the negative consequences of UV radiation. Keep your exposure to midday sun brief and wear sunscreen and sunglasses as needed. Wearing sunscreen does reduce vitamin D synthesis in the skin. However, a good strategy may be to wear sunscreen on the face only and limit exposure to direct midday sunlight so that you don't get sunburn in those parts of your body that are not covered by sunscreen. Well, how long would you need to be in the sun to produce enough vitamin D to meet your daily requirements? The answer to that question depends, of course, on the amount of UV radiation your location gets at that time, and also on variables such as your skin color and how much clothing you wear. I've left a link in the description box below in which you can experiment with the different variables a little bit yourself. And the system will tell you how long you would need to be outside to make 1000 international units of vitamin D, assuming that about 25% of your skin is exposed to the sun. 25% of your skin is equivalent to your face, neck, arms, and hands. To give you an idea, imagine someone with white skin living in Boston, Massachusetts, such as Jim here. Jim goes out into the midday sun on a clear day in August. Under these conditions, it would take him around 12 minutes to make about 1000 international units. In January, Jim would not be able to make any vitamin D in Boston. That has to do with the angle of the sun and the strength of UV radiation reaching Boston in the winter, which is simply insufficient for meaningful vitamin D synthesis. By contrast, if Jim lived in Miami, Florida, he would need about 10 minutes of midday sun at any time of the year. That is simply because Miami is closer to the equator and UV irradiation is much less seasonal there than in the northern parts of the United States. If someone has darker skin or wears more clothing or it's a little cloudy, then more time in the sun would be needed to make these 1000 international units of vitamin D. That is also why, depending on a large number of factors, such as where you live, your age, the color of your skin, the amount of clothes you wear, you may require supplemental vitamin D. Again, this will be covered in detail in a separate video. However, it is also important to understand that increased vitamin D synthesis is not the only beneficial effect of sunlight exposure on blood sugar regulation. This means that even if it helps you avoid vitamin D deficiency, taking a vitamin D supplement is not the same thing as being in the sun regularly. I actually recommend a supplement for most people at least, and I also recommend being in the sun regularly. That has to do with the other two reasons that sunlight exposure can be beneficial for our health in general and glucose tolerance in particular. Sunlight and glucose tolerance, avoidance of circadian disruption. The second mechanism through which light affects our glucose tolerance is that light helps us regulate our circadian rhythms. What does circadian rhythm even mean? Well, all biological processes are to some degree regulated in a circadian fashion, meaning that they change in a roughly 24-hour cycle. An obvious one is that we get tired and need to sleep at some point during a 24-hour period. But our circadian rhythm is about a lot more than sleeping and being awake. Basically, every process in every cell is to some degree regulated by our circadian rhythm. And that circadian rhythm is mostly established by two primary inputs: our exposure to light and when we eat. That food intake regulates what is going on in a cell makes sense, because cells can potentially directly sense an increase in nutrients in the blood and respond to them, right? But what about light? How can our cells see whether it's day or night? Well, mostly we could argue cells can't, even though I'm going to get to this later in this video. But each cell also has a clock. No kidding here. There are a number of proteins in each cell, the concentration of which ebbs and flows roughly in a 24-hour cycle. And through the concentrations of these proteins, the cell can tell what time of day it is. These individual cell clocks are set by a master clock in a part of the brain called the suprachiasmatic nucleus, or SCN. And that master clock is kept in alignment with the outside light by receiving information about ambient light directly from the eyes. Let's talk briefly about how this works. This here is a cross-sectional view through a human head and brain. In the brain, we have two areas that are most relevant for our circadian rhythm: the SCN and the pineal gland. That's the gland that secretes melatonin, the hormone that helps us sleep. When our eyes sense bright light, and particularly light in the blue part of the spectrum, this stimulates the production of a protein called melanopsin. Melanopsin has two major functions. First, it conveys to the SCN that it is day, which is of key importance in maintaining our normal circadian rhythm. And second, melanopsin suppresses the production of melatonin in the pineal gland. As a result, the concentration of melatonin in the brain remains low as long as we're exposed to bright and particularly blue light. And at night, when ideally no light hits our eyes, no melanopsin is formed, and this allows melatonin to be produced in the pineal gland, which then in turn helps us sleep through the night. We speak about circadian disruption or circadian misalignment if there's a disconnect between our internal clocks and our external environment, such as the light we receive through our eyes or the time of day during which we eat. Or if there is a misalignment between our master clock in the brain and the individual clocks in all of the tissues of the body. And any such misalignment can make it more difficult for the body to regulate all of its functions, such as its sugar metabolism. Under ideal conditions, think caveman and cavewoman before the invention of fire, food intake occurred only when there was daylight. That was very predictable. Nutrients such as glucose, fat, and all of the micronutrients were coming in during the day at the same time that the eyes and the master clock conveyed to the body cells that it was daytime. All of the processes that were needed to deal with incoming nutrients were switched on in all of the tissues that had to deal with these nutrients: the liver, the pancreas, the muscle, the kidneys, etc. And vice versa, when it was dark, there was reliably no food coming into the body, and the cells could focus on other things, such as DNA repair or autophagy. A reliable fasting period overnight was also perfect to help cells deal with nutrients that may have accumulated overnight. For example, if the caveman or cavewoman found a large patch of berries and ate ripe berries all day, the fructose from all of these berries would have been taken up from the blood by the liver and converted to fat during the day. This would have slightly increased the fat content in the liver, which the liver can handle as long as it isn't too much. And it wouldn't ever be too much, because during the long fasting period overnight, the liver cells could burn this fat or get rid of it by transporting it to the fat tissue. That long fasting period overnight, during which historical humans rested and were not exposed to light, or at least not much light, was also a perfect time for the liver and other tissues to increase repair and maintenance processes. Considering that humans almost certainly evolved in this type of environment, with light and food intake during the day and mostly darkness and no food intake during the night, you can probably appreciate that most of us live very differently today. Now, this example may sound as if avoidance of food intake at night matters a lot for optimal regulation of our metabolism. And that is correct. As we discussed in 2 prior videos about time-restricted eating, where I explained that our glucose tolerance tends to improve if we shorten our eating window and eat mostly in the early part of the day, during daytime. However, we have good evidence that our exposure to light matters just as much. For one, we do have evidence that too little bright sunlight during the day is associated with insulin resistance, glucose intolerance, and an increased risk for type 2 diabetes. And independent of that, we also have lots of data showing that insulin resistance, glucose intolerance, and a higher risk of type 2 diabetes are associated with exposure to artificial light at night. Now, what is meant with this artificial light at night? Does this mean we cannot use any electric light in the evening at all? No, I wouldn't go so far. Looking at this literature, it seems to me that there are two things specifically we want to pay attention to. First, we ideally want to sleep in complete darkness. And second, we may want to avoid exposure to bright lights and specifically to blue light in the maybe 1 to 2 hours before going to sleep. This is because bright white light that is rich in the blue portion of the visible spectrum, that's right here with a wavelength of around 445 to 480 nanometers, is very effective at suppressing the production of melatonin in the pineal gland. Again, the pineal gland sits in the middle of the brain, and producing melatonin there is of critical importance for us to be able to sleep through the night. So ideally, in the later part of the evening, we would dim the lights and shift the color spectrum towards the orange and red portion of visible light. I'll talk later about how this can be done. For now, let's focus on why a lack of bright sunlight during the day or artificial light at night could lead to glucose intolerance. I'd say that we don't have a totally clear answer to that. But let's discuss a few hypotheses. For one, both a lack of bright light and artificial light at night have been connected to sleep issues, both in terms of too little sleep and also in terms of poor sleep quality. In clinical experiments, as little as 1 or 2 nights with too little sleep substantially increases insulin resistance. So I'd say that this is one likely link there. Second, people who get more light during the day may have a better vitamin D status. And as I explained earlier, that could play a role here. Third, people who are exposed to more artificial light at night because they watch TV or play video games late into the night or because they do shift work may be more likely to eat at night. The observational studies that have linked more artificial light at night to an increased risk of type 2 diabetes have not always been able to adjust for food intake at night. In other words, food intake may be a confounder here, suggesting that, at least to some degree, the association between artificial light at night and insulin resistance and glucose intolerance may be explained by food intake at night or a shorter fasting period overnight. Now, one thing that has remained unclear is whether vitamin D deficiency, sleep deprivation, and eating food at night completely explain why a lack of bright sunlight during the day and artificial light at night are associated with insulin resistance, glucose intolerance, and an increased risk of type 2 diabetes. The data we currently have doesn't allow us to answer that question. I'd say it's possible that there is more going on though, and that specifically a lack of bright sunlight and artificial light at night could by themselves reduce our body's ability to maintain normal glucose levels just because the optimal circadian rhythm is disrupted and the interplay between the different organs that maintain glucose homeostasis is not as smooth as it could ideally be. Lots of research is still to be done in this area, but for now, I'd say that we do have enough evidence to strongly suggest that our glucose tolerance will benefit from a lifestyle in which we aim to maintain a normal circadian rhythm. Sunlight and glucose tolerance, improved mitochondrial function. And this brings us to the third mechanism through which light affects glucose tolerance, and that has to do with near-infrared light. Now, this is a really exciting area of research. Near-infrared light, which like ultraviolet light is not visible to us, accounts for 52 to 55% of all light photons coming from the sun that hit our bodies if we're outside. The near-infrared light is a huge amount of light energy, and in my opinion, it would make a lot of sense that our bodies evolved to use this somehow. And yes, there is indeed some really intriguing research that strongly suggests that the red light of the visible light spectrum, as well as the near-infrared light, play an important role in maintaining optimal mitochondrial function. Well, the first and obvious question is, how can this be? Most of the cells in the human body never see light, right? Well, that is not correct. Ultraviolet light does not penetrate the body and is instead entirely absorbed in the very top layer of the skin, in a layer that is maybe 1/10 of a millimeter thick, about the thickness of hair. That's why UV light is potentially so damaging, because all of its energy needs to be dealt with by a very thin layer of cells. Similar for the blue and green and yellow portions of visible light, which also do not penetrate deeper into the skin. Red light goes a little deeper. See what happens if you shine a flashlight at your hand. You can see some light shining through, and the light that shines through is all red. That is because only the red portion of visible light can pass through the tissue deep enough to come out on the other side. Now, near-infrared light can pass even deeper into the body, based on some data 8 to 10 centimeters deep. That's 3 to 4 inches. That means that 100% of all cells in children will receive near-infrared light if they are in the sun, and still about 70% of all cells in adults. A substantially smaller portion in adults who carry extra body fat, by the way. Near-infrared light can even penetrate the skull and reach the brain. Bones are not much of an obstacle for near-infrared light. So yes, if we're exposed to red and near-infrared light, such as from the sun, the light will reach many or even most of the cells in our body. And what does that now have to do with mitochondria and glucose tolerance? Well, remember that mitochondria are the power plants of each cell. Fuel such as glucose or fatty acids can be burned here, just like coal is burned in a coal power plant. Under the influence of oxygen, O2 here, the mitochondria make ATP and carbon dioxide, CO2. ATP is a chemical form of energy that the cell uses for many different processes. So this isn't just to power your muscle contractions and movements. ATP is a very big deal for most biological processes inside of our cells. Now the interesting part. Mitochondria that are exposed to red or near-infrared light work more efficiently than those that are not exposed to red or near-infrared light. That means that per unit of time, they burn more glucose and fatty acids and produce more ATP. This is a fairly new area of science, and we need a lot more research to really know what's going on here. But we have some data already about potential mechanisms through which red and near-infrared light may affect the efficiency of ATP production by the mitochondria. The first potential mechanism is that red and near-infrared light acts directly on one of the key enzymes in the mitochondria that convert fuel to ATP. This enzyme is called cytochrome C oxidase, and its responsiveness to light is in its name. Cytochromes are what we call chromophores. They absorb light of a specific wavelength. In the case of cytochrome C oxidase, near-infrared light increases the activity of the enzyme. The second potential mechanism is that red and near-infrared light may reduce the viscosity of the water within mitochondria. Water with reduced viscosity is smoother, providing less resistance, so this would allow all of the molecules to move around more easily. Again, this is expected to speed up the process of making ATP and make it more efficient. Third and lastly, there are some data that suggest that near-infrared light may stimulate the expression of melatonin within the mitochondria. This is really confusing. We mentioned earlier that blue light hitting the eyes leads to a suppression of melatonin production in the pineal gland in the brain, and that blue light therefore keeps us awake. Now, what I'm saying here is that elsewhere in the body, within the mitochondria of each cell, another part of light, namely near-infrared light, increases the production of melatonin. Yes, that at least is one hypothesis in the field based on strongly suggestive data. Now, why is this important here? Well, it's highly relevant because the production of ATP by the mitochondria is a tricky process because oxygen is involved. Every once in a while, something goes wrong in the process of burning fuel in the mitochondria, and some of the oxygen molecules form what we call reactive oxygen species, or ROS. ROS are highly reactive molecules that can damage the entire machinery of the mitochondria. To stick with the power plant analogy, imagine an accident in the power plant, a major fire breaking out, and some or all of the machines and instruments being damaged. So, how do we prevent this? A power plant may have some firefighters, and mitochondria have antioxidants to deal with the reactive oxygen species. And melatonin is one of the most powerful antioxidants known to us. Okay, to summarize, mitochondria are more effective at converting fuel, such as glucose and fatty acids, to chemical energy, ATP, if they are exposed to red or near-infrared light. The mechanisms seem to involve a direct effect on one of the enzymes involved in the energy production process, cytochrome C oxidase, by lowering the viscosity of water in the mitochondria and by increasing the production of a key antioxidant, melatonin. Okay, so how is all of this relevant for glucose tolerance? Well, one prediction would be that if red or near-infrared light really makes the burning of glucose and fat to ATP more efficient, then this should A. increase ATP synthesis, B. increase the uptake of glucose from the blood into these cells, and C. increase the amount of CO2 that is exhaled. One team tested this hypothesis. In a randomized controlled study, they shown red light on the backs of people before they had an oral glucose tolerance test, or OGTT. And on another day, they repeated the process, but unbeknownst to the participants, did not shine the red light onto their backs. This was the placebo control. And take a look at what they found. The increase in blood glucose levels in response to drinking the 75-gram glucose beverage was a lot smaller in participants after they had been exposed to the red light. In other words, their glucose tolerance was substantially improved. The increase in blood glucose above the baseline was a whopping 28% lower after exposure to the red light. And not only that, they also exhaled measurably more CO2, as the model would predict. Now, it's important that we don't get carried away too much here. This is one study, and even though it confirmed a hypothesis that was based on prior research, it's just one study. This needs to be confirmed, and in general, there's a lot more to learn about this. It is, for example, unclear whether this effect can be triggered only by a high dose of red light of a very specific wavelength, as in this study, or whether this would translate to exposure to normal sunlight. However, I'd say that there is reason to be cautiously optimistic that this line of work could give us a better understanding of the factors involved in the development of glucose intolerance and type 2 diabetes, and certainly hold some promise for interventions to improve the condition. These data are certainly also consistent with the data showing better glucose tolerance and a lower risk of type 2 diabetes in people who spend more time in natural sunlight. So in addition to the fact that more sunlight improves our vitamin D status and our regulation of a natural circadian rhythm, this could be an alternative mechanism explaining this association between sunlight exposure and a lower risk of type 2 diabetes. Now, one particular area of research that I would really like to see in the future is how regular exposure to near-infrared light, or a lack of exposure to near-infrared light, may affect the two key factors that determine glucose tolerance: insulin sensitivity and beta cell function. I'm saying this because for years there have been numerous publications that have described that in pancreatic beta cells that become dysfunctional, a key cellular observation has been mitochondrial dysfunction. And similarly, in liver and muscle cells, a common observation associated with insulin resistance in these tissues has been mitochondrial dysfunction. I'm not suggesting that a lack of near-infrared light is the only cause of mitochondrial dysfunction. However, it could be one contributing factor. Well, how may all of this be relevant for you? It is relevant because most of us are now living in an environment that is devoid of near-infrared light. Take a look at the spectrum of sunlight. As mentioned earlier, about 52 to 55% and maybe more of the energy coming from the sun is near-infrared light. This varies a bit from morning to midday and evening, but in all cases, much of the light that hits us when we're outside is near-infrared light. Contrast this to today. If we're indoors and use energy-efficient lighting, such as LEDs, these emit only light in the visible spectrum. In contrast, the old energy-inefficient incandescent light bulbs produced a lot of near-infrared and infrared light, which is also why they became hot to the touch and were so inefficient. Then add to this that infrared light from the sun used to penetrate simple single-pane windows. With energy-efficient glass in modern buildings, that is no longer the case. This modern glass is designed to keep warmth, which is essentially infrared radiation, inside. And the side effect of that is that near-infrared and also infrared radiation from the sun are kept out. As a result, you may not be exposed to any near-infrared light in your home, your office, your factory floor, your gym, or any other modern indoor environment. And if this is indeed an essential nutrient, that would almost certainly not be ideal. Now, I'm not suggesting we abandon energy-efficient lighting or switch back to single-pane glass windows. However, I do anticipate that the research I've shared here will lead to technological advances that help us create energy-efficient buildings that also provide healthier light for the people living in them. Some LEDs are already available that emit near-infrared light and that can be dimmed or programmed to limit blue light at night. But these are quite expensive still and hard to find. Summary, conclusions, and suggestions. To summarize, our exposure to light can affect glucose tolerance and its key determinants, insulin sensitivity and beta cell function. Potential mechanisms through which light affects glucose tolerance include that first, UV light triggers vitamin D synthesis in the skin, which helps us avoid vitamin D deficiency. Second, exposure to bright sunlight during the day and the lack of bright and particularly blue light at night improve our circadian rhythm and, linked to this, our sleep. And third, exposure to red and near-infrared light improves mitochondrial function. Now, one very important thing to understand here is that I'm not suggesting that exposure to light is a cure-all kind of thing. What I'm suggesting is that our lack of exposure to sunlight or exposure to blue light at night could be one of the potential causes or contributors to your glucose intolerance. And if that is the case, then improving your relationship to light may well be one step that can help you normalize your glucose tolerance. So what does the science suggest that we can do about this specifically? I can see 4 actionable items. First, make sure you are not vitamin D deficient. Have your plasma 25-hydroxy vitamin D level measured and make sure it's above 20 nanograms per milliliter at all times. In my opinion, ideally you'd aim for the higher end of the normal range, somewhere between 30 and 50 nanograms per milliliter. If you are vitamin D deficient, make sure you get some direct sunlight every day and or take a supplement. Second, make sure to go outside every day during daylight hours. Keep exposure to strong midday sun to a few minutes and wear sunscreen and sunglasses as needed during that time. But other than that, I'd encourage you to reconsider your relationship with the sun. Try to find ways to be outdoors more. Spending time in shady green spaces such as parks, forests, or your backyard is also a great idea. Near-infrared light reflects off many surfaces, particularly green surfaces, and so you'll be exposed to near-infrared light to some degree even under the canopy of a tree or sitting in the shade in your backyard. And even a cloudy day is usually brighter than the light we get indoors. And it's certainly a much wider spectrum light than what our indoors lighting usually provides. Clouds filter out some of the near-infrared light, but I would argue that particularly when near-infrared light is hard to get, is it probably important to spend some time outdoors. Third, sleep in an entirely dark room. Use blackout shades in your bedroom and remove or cover any light source, including night lights or the display of an alarm clock. And if you need to visit the bathroom, use as little light as possible. And fourth, keep your exposure to artificial light, particularly blue light, limited after the sun has set, particularly in these 1 to 2 hours before you go to sleep. Use software that filters out blue light on your electronic screens, such as your smartphone, tablet, computer, or TV, and use mostly dim lighting after the sun has set. One objective here is to help you avoid circadian disruption. My strong suggestion is to combine these suggestions with a form of time-restricted eating, ideally early time-restricted eating, and your chances are good that your body will return to its natural circadian rhythm. Alrighty, that's it for this video. If you like this, I would appreciate it if you left me a thumbs up below, and particularly if you shared this video with anyone you think may be interested. As always, I have published a separate blog post that discusses the science in more detail and that provides links to all of the scientific references. It is linked in the description box below. Before we close, I would like to invite you to join the Nourished by Science Prevention Program. In this program, I work with people directly who want to learn about and implement dietary and lifestyle habits that minimize all of the root causes of chronic disease. We choose different areas of emphasis to work on at a time, be it blood sugar, blood lipids, blood pressure, body weight, or micronutrient adequacy. We spend some time learning about a specific risk factor, assess to which degree we are exposed to it, and then take guided steps together to address whatever issues we personally may have. We regularly hold live calls to discuss the scientific evidence and practical steps that program members can take, and we're always available to answer questions. If this sounds like something you may be interested in, you can find a link below to get more information. And with that, I would like to close by thanking the first patrons of the channel. Through patreon.com, you can support the channel directly and also connect more closely with me. We have a community space where you can ask me questions, where I occasionally share updates about what I'm working on, and where you have the opportunity to join me in live group calls to discuss the science covered in each video in more detail. If you appreciate the fact that I make nuanced, evidence-based content freely available here on YouTube and on my blog, I would be honored if you would consider becoming a patron as well. Thank you so much, and take care.